Hi, good afternoon and good morning. Thank you for attending our webinar, Unlocking the Hidden Dimensions of Biology, Integrating 10x Genomics Single Cell and Spatial Profiling with Oxford Nanopore Long-Read Sequencing. My name is Corey Asafano, and I'm with 10x Genomics, and I'm going to be moderating today's webinar. Today, we have several great speakers. Carolyn Peterson, 10x Genomics Science and Technology Advisor, will be giving us an overview first on our Chromium single cell and Visium platforms, as well as also providing some insights to why long-read sequencing might be the next best fit for your next spatial or single cell project. Additionally, she'll also be providing some application highlights as well. Following Carolyn, Daniel Giraldi, Associate Director of Technology Markets from Oxford Nanopore, will be providing an overview on the technology that powers Oxford Nanopore's long-read sequencing. Our guest speaker today, Anushka Jalakar, postdoctoral researcher from New York Genome Center, will be providing us with a deep insight into her research on how the combination of 10x Genomics and Oxford Nanopore have enabled her to answer several very unique biological questions. I'm going to turn everything over to Carolyn now to kick things off. Thank you.
Good afternoon, everyone. My name is Carolyn Peterson, and I am a Science and Technology Advisor from 10x Genomics. I'm excited to have the chance to talk about long-read sequencing today and how you can apply Oxford Nanopore sequencing to 10x Genomics assays. I'll begin today with a brief overview of the 10x Genomics platforms. It goes without saying that the field of genomics is fast-paced and rapidly evolving, and 10x has evolved with it, with our new technologies that help better resolve the complexity of biology. Over the past decade, single-cell RNA-seq has become instrumental in characterizing the heterogeneous response arising from specific cell types and states. More recently, the emergence of spatial transcriptomics has allowed researchers to generate spatially resolved data types while retaining tissue to architecture to get a clear picture into what's going on inside intact tissues. Xenium, our newest platform, is our microscopy-based in situ platform that enables gene detection at the subcellular resolution. So now you can investigate what's going on within individual cells. Together, the three platforms at 10x Genomics are multifaceted and complementary to each other, enabling powerful insights into biology with unparalleled resolution. Today, I'll be focusing primarily on our Chromium single cell and Visium spatial platforms, as these are our sequencing-based assays that are compatible with long reads. Over the last couple of years, we've seen an increasing amount of interest in long reads, with LRS being named Method of the Year for 2022 by Nature Methods. At 10x, we want to enable you to explore new applications with our technologies, so we've partnered with Oxford Nanopore to demonstrate that Chromium and Visium libraries can be used to interrogate full-length transcriptomes for isoform discovery and characterization. Chromium and Visium were originally built for short-read sequencing, but new assays and kits available from Oxford Nanopore allow you to preserve the full-length cDNA during library construction in order to sequence the entire transcript. Now, you might be wondering why researchers want to use long reads to begin with. And the answer is the ability to capture even more information from single cells or tissues, such as alternative splicing patterns and novel isoforms. Alternative splicing is the biological process in which more than one mRNA or isoforms are generated from the transcript of the same gene through different combinations of exons and introns. It is an important evolutionary mechanism that expands the proteome by increasing the functional diversity of gene products. Now, even though different isoforms from the same gene can have highly similar sequences, they may have very different biological functions. Isoforms have been found to play a critical in development, and many diseases, including cancer, have been known to exhibit isoform switching. So it is critical to not only characterize the different isoforms, but also understand how they function, as well as how they are organized spatially within tissues. Traditional short-read sequencing can leave certain types of information, like isoforms, invisible to researchers. Short reads only sequence 90 base pairs of the cDNA, and during library construction, you must fragment your full-length cDNA transcripts so that they are compatible with the sequencers. Long-read sequencing, on the other hand, is a method by which you sequence your transcript molecules in full, making it possible to not only identify and characterize isoforms, but also perform quantification on the gene and isoform level. In this paper from the Wellcome Sanger Institute, they illustrate nicely how researchers can couple long-read sequencing with 10x to get a better understanding of the role of isoforms in disease. Here they were studying acute lymphoblastic leukemia, a devastating malignancy in infants. About 70% of ALL cases are known to be caused by rearrangements of the oncogene KMT2A. For this experiment, they used a multi-omic approach, utilizing both single-cell RNA-seq and ATAC-seq to understand the transcriptomic and epigenomic changes underlying this disease. They also used long-read sequencing on the RNA-seq libraries in order to characterize fusion isoforms of the KMT2A gene. With their resulting data, they were able to identify a rare blast population, which only made up 1.5% of the total cell population, but 80% of these cells came from patients under 6 months of age. These cells exhibited transcriptional and epigenomic signatures similar to HSPC cells, though they also exhibited leukemic signatures. With their long reads data, they were able to confirm the malignancy of these cells with the significantly elevated levels of fusion reads of the KMT2A gene that they found. They also discovered that these cells had higher expression of genes relating to apoptosis resistance and ribosome biogenesis, suggesting that these cells play a role in drug resistance and relapse. Overall, by using a multi-omic approach with long-read sequencing, they were able to better understand the mechanisms of infant ALL, and their findings have direct clinical implications by potentially identifying new therapeutic targets for treating this disease. So now that we have a better understanding as to why long-read sequencing with 10x can be so important, let's quickly review just how the 10x technology actually works. So the 10x single-cell portfolio consists of 4 main applications: 3' gene expression, 5' immune profiling, multiome for RNA-seq and ATAC-seq, and flex for fixed or FFPE samples. Today, we'll focus on 3' and 5', as these assays both generate full-length cDNA. The general workflow remains the same for all single-cell assays. We use advanced microfluidics to partition cells into single-cell droplets that also contain a barcoded gel bead. The main difference between the assays is the capture sequence on the bead oligos. Both 3' and 5' have oligos containing a 10x cell identifier barcode, a unique molecular identifier, and a capture sequence. 3' uses a poly-DT sequence to capture the 3' end of the mRNA, and 5' uses the template switching oligo to capture the mRNA by the 5' end during reverse transcription. Now, for our Visium spatial platform, we have 2 different workflows available. First, we have the direct placement method for fresh frozen tissues, in which you place your tissue section directly onto the Visium slide. And then we have the site-assessed method for fresh frozen, fixed frozen, or FFPE samples, which uses instrumentation and a probe-based approach to capture gene expression measurements. Today, we will focus on the direct placement method, as this captures full-length transcripts necessary for long reads. The hallmark behind the Visium technology is the gene expression slide, which contains 4 capture areas, each arrayed with thousands of barcoded spots. With the direct placement method, you'll place tissue section directly onto the capture area, and when you permeabilize your tissue, the transcripts will be captured and barcoded by the oligos found in each spot. Similar to our 3' assay, the oligos contain a poly-DT capture sequence and a UMI. But instead of a cell barcode, there is a spatial barcode that correlates to an XY coordinate back on that original capture area, which retains the spatial context when we capture the transcripts. As you can see here, 3', 5', and Visium workflows are capturing off the poly A tail and use reverse transcription to tag the cell or spatial barcode and UMI onto the transcript. Because we are capturing by the poly A tail, this process is reverse transcribing the full-length transcripts, which is a critical input into long reads. Now, the 10x library construction workflow, as I mentioned before, were designed for short read sequencing. And one of the first steps is fragmenting your cDNA. So when you want to detect isoforms, you want to avoid this step altogether in order to retain that full-length cDNA and your isoform information. So instead, you'll follow the 10x workflow up until cDNA amplification and cleanup, but then you'll use ONTs kits to prepare your cDNA for sequencing. And with long reads, you're sequencing the full-length cDNA, which will enable you to detect isoforms or fusion events splice variants, and even single nucleotide variants. To test the compatibility and reliability of our single-cell and spatial assays with long reads, 10x collaborated with ONT to run a validation experiment with 3', 5', and Visium sequenced side by side with Illumina short reads and ONT long reads. You can follow the QR code on the right to get direct access to the tech note describing the experimental design and results further. But in short, they were able to confirm that long read and short read datasets produce highly concordant results, confirming that your biological insights will remain consistent when you switch to long reads. However, with long reads, you are getting even more information from your cells, and that's the isoform information. From the resulting datasets, you can see that both sequencing platforms had similar clustering results, and the same cell populations were detected in similar abundances. There's also high consistency in terms of the genes and UMIs detected per cell, as well as highly concordant gene expression profiles and cell calling. Next, they looked at the expression of the PTPRC gene, which plays an important role in immune cell receptor signaling. With the short read RNA-seq data, you can see that this gene is expressed in the majority of the cell populations. However, with long reads, we are able to distinguish the different isoforms of this gene, and we can see that isoform expression is cluster dependent, in other words, cell dependent, which further supports the fact that different isoforms have different roles among cell populations. Finally, I want to wrap up my portion of the talk by showing this final application highlight, which actually comes from today's guest speaker, Anushka. In this paper, they were interested in creating a single cell and spatial map of the splicing variation across brain regions of the mouse. In the hippocampus and prefrontal cortex, multiple cell types undergo differentiation, which is influenced by development-specific splicing. So they wanted to get a better understanding of how and where this occurred. With their single-cell long-reads data, they were able to identify several different isoforms. One in particular was for the NSMF gene, a synaptic gene involved in memory formation. And they found that the different isoforms were preferentially expressed among different cell types. In particular, the isoform that lacked a specific 6-nucleotide microexon, shown by the blue arrow, was completely absent in neuronal cells, suggesting that this tiny microexon actually played a critical regulatory role in neuronal functions. They also used Visium with long reads to get a spatial context to isoform expression. Strikingly, they found that SNAP25, another synaptic gene, underwent isoform switching from SNAP25A to B in a posterior to anterior gradient, which is illustrated nicely in the Visium data shown on the right. This data supports the idea that the microenvironment can dictate brain region-specific splicing for some genes. In summary, this work highlights the advantage of combining single-cell and spatial with long reads, and how characterizing differential isoform expression across cell types and tissue regions are critical to better understanding brain development. And I'm personally excited to hear more about Anushka's recent work using long reads later in this webinar. With that, I'd like to wrap up my portion of the talk by highlighting the complementarity of the 10x toolkit. Each technology platform that we build lends a unique perspective and an overall biological picture, spatial, cellular, and subcellular. Every assay within each platform is uniquely tailored for generating powerful data sets that will lead to meaningful and insightful discoveries. To close, I thank you for your time today, and I'll now pass it on to Daniel to talk more about Oxford Nanopore sequencing technologies.
Wonderful. Thank you, Carolyn. Let me share my screen here. And thanks to each of you who joined today. I'm also looking forward to Anushka's presentation. Okay, I think this is right. Are you guys able to see that? Okay. Carolyn, thanks again. That was a great overview of single cell and 10x genomics, and also a lot of the applications with Oxford Nanopore. So I'm just going to do a quick presentation and cover some of the perspectives from the sequencing side and also our workflow, and then we'll get to hear from Anushka. So when we think about single cell sequencing from single cell transcriptomics from the sequencing perspective, single cell transcriptomics has been around for a long time, for many years, but for a long time it was very low throughput and it was manual methods. It's really only been the last few years that we've seen exponential growth in the number of samples across many research areas. And I think that's in large part been driven by the accessibility of the 10x Genomics platform, which makes it easy to analyze tens of thousands of cells. And the field has realized that having many more cells in the analysis gives you much more sensitivity to rare cell types and allows you to assess the biological diversity and complexity of these samples. So now that we have these platforms that make it easy to analyze so many cells, it's no longer seems like a crazy intractable idea to have Atlas scale projects with over 1 million cells. And as we've added more and more cells to the experiment, we've added resolution, as I mentioned, but there's still types of biology that we cannot resolve by adding more cells. And Carolyn hinted at that, which is anything that happens outside of the 3' or 5' tag is invisible when we sequence it on a short read platform. So here we just have a single gene annotation in the top panel. In the middle panel, you see a typical short read library, as you'd expect, with those reads aligned at the 3' or 5' end, depending on the 10x genomics kit that you're using. And anything that happens outside of that region is missing. If that's splicing information, it can be mutations. And in the bottom panel, we have the Oxford Nanopore read. So you can see each of those reads is a full isoform, and it's got the exon-exon connectivity contained within it. And it also captures any other biological diversity that's carried into the transcript. So for many applications, that information, that 80% of the transcript that's missing, does matter. And I'd encourage you to ask the question for the different projects that you're doing. Do these things matter to me? Does alternative splicing matter, or isoform switching events? Does it matter being able to identify transcripts that are targeted by NMD, nonsense-mediated decay? And this is one of the reasons that protein levels diverge from gene expression levels when you're just using a short read at the end of the molecule and can't identify this. In terms of discovery, there's some advantages of having full-length reads and doing CRISPR screens, where you can actually identify whether or not a mutation is carried into the transcript via CRISPR for a particular cell. You get a multimodal readout of genotype plus expression, and you could get complete TCR and BCR sequences and better isotype assignment. In disease targets, particularly cancer, you have things like fusion gene identification. Often fusions happen, and Carolyn, I think, showed an example away from the ends of the transcript. You can identify those with long reads. The ability to do targeting of your panels when you're sequencing on Oxford Nanopore, and looking at isoform QTL. I'm only going to talk about 2 publications briefly. On the left, you see now a single cell, in this case, a t-SNE plot that looks very similar to what you would typically see, but this is a single cell clustering done at the isoform level. And what we're starting to see in some of these publications is you start seeing additional cell subtype clusters when you use the isoform information, because you're getting more information about what each individual cell is actually expressing. On the right, you see the 10x Genomics Visium. And I think actually this is a similar gene that Anushka, the paper was looking at. And you can see there's this dramatic difference in this particular gene, which isoforms used in the midbrain. There's almost no expression of the 202 isoform in the midbrain. This is something that is invisible when you're using short read sequencing and you only have the gene level expression. Again, in mouse brain, from mouse brain single cell, you can see here an isoform switching event where you have strong expression of the CLTA206 isoform on the right in these precursor cells. And the 204 isoform is very dominant in the mature neuronal cells. And in this publication, the authors found 76 genes in the mouse brain that have this type of isoform switching across 2 different cell type clusters. Another really important piece of information for many studies, especially in cancer, is being able to identify not just the splicing, but mutational changes. There's a lot of mutational burden in cancer cells, and being able to read those across the full-length transcript is incredibly powerful. On the left in the figure here from Ruli Gao's group at Northwestern University, you see on the x-axis single cells, And on the y-axis, you see different mutations. And the difference here is that when you're using long reads, you can pick up mutations anywhere across that gene body, anything that's transcribed into RNA. And you're not limited to just the tag at the 3' or 5' end. You can see just from this plot at the high level view difference between the the mutational burden in the tumor cells versus the wild type cells. And you can even see a little bit of clonality if you look closely here. Now, not only do you have that mutational information on the right, you also compare that directly with splicing changes. And you can see here some examples of genes that have dramatically different isoform usage in the normal cell versus the tumor cell, and myeloid versus T and natural killer cells. So the last slide is just to summarize that we have a simple 10x Genomics certified compatible workflow to take 10x Genomics single cell or Visium samples. Those are prepped exactly per the 10x Genomics protocol. And we have a very fast and simple library prep. So it takes about 3 hours to go from your 10x Genomics cDNA to a library that's ready to load onto a sequencer. Our Promethean device gives you around 80 million cell and barcode assigned reads per flow cell. And those devices can accommodate up to 48 different flow cells in parallel. And then we have an analysis solution that gives you your gene and isoform count matrices, like you would typically get out of Cell Ranger, and then you would do further downstream analysis. Thank you very much. And I'd like to pass back to Corey to pass on for Anushka. We're looking forward to your talk. Thank you.
Thank you, Daniel. Our next speaker, Anushka Jalakar, is going to go through her research and how the marriage of these two technologies have enabled her to provide very unique biological insights to a very unique sample type. And I'm going to turn it over to her.
Can everyone see my screen and hear me okay? All right, so today I'm going to be talking to you about a spatiotemporal role of alternative splicing in the mammalian brain. Like Corey mentioned in the beginning, I am a postdoc at the New York Genome Center, but today's talk is going to be about my graduate work from the Tilgner Lab. So I don't need to go over most of my introduction about what splicing is, because the previous two speakers really did a great job covering all my bases, but let's just go through a quick overview just in case. So we all know that the central dogma says that we go from DNA to RNA in a process called transcription, and then we go from RNA to protein in a process called translation. But a lot of times what is ignored is that there is a pre-processing step before translation, known as splicing, which is mediated by a complex protein structure called the spliceosome. And along with taking out the introns, which are part of the junk DNA, it also takes out some exons, which means that the same mRNA molecule leads to multiple mRNA isoforms, which are then translated into distinct proteins. And that leads to proteome expansion. So the worm, the mouse, and human, we all have the same number of genes, but the number of isoforms per gene varies considerably. So this is kind of nature's way of combinatorially expanding our proteome and making us, quote unquote, more complex, whatever that means. And if this hasn't convinced you that it's important, another reason why splicing is really important is because, say, there's a gene called CD33, which is an immune gene. And depending on whether you have or don't have an exon, which encodes a particular domain in the protein, it can make the difference between health and disease. In this case, it is neurotoxic in terms of Alzheimer's disease, and the isoform without the exon is neuroprotective. So isoforms can be tissue specific, meaning that you can have the same gene having two different isoforms in, say, the gut and the brain, and that leads to further downstream consequences, depending on what other genes they interact with in their network. Isoforms can be region specific, meaning that the front and the back of the brain can have different isoforms. And to make matters a little bit more complicated, they can also be cell type specific. So I'm mostly concerned about the brain, but of course this can be extended to any other tissue. And the brain contains more than just neurons. It has a vast diversity of different cell types. And depending on which cell type you look at, the isoform repertoire is very different. So the best way to study anything on a cell type specific level is to do droplet-based single-cell RNA-seq. And we've just listened to Caroline talk about the different 10x offerings. But like she already mentioned, this is a 3' font method, and we lose a lot of crucial information. So just to illustrate what I mean, the cartoon I was showing you earlier showed that you have different protein isoforms from your RNA and cDNA, but when you sequence the 3' end of your transcript, you can maybe tell the difference between protein C versus the other two, but you really can't distinguish between protein A versus B. And the best way to try and do that is by doing long-read sequencing. So the way we did this is we came up with a technique called single-cell isoform RNA sequencing, or scissor-seq, which is what today's webinar is all about technically. And the first half of the protocol is the exact same as your regular 10x, but pre-fragmentation, we actually save our cDNA and we send it off for long-read sequencing. What that means is because it's the same exact library with the same exact barcodes, you can deconvolve the barcodes from every long read, trace it back to the cell of origin, and get cell type resolved isoforms. I do want to emphasize that with 10x, say you have this example isoform with the barcode on the 3' end, you will be sequencing part of it with a sequencing technique that maybe doesn't have the 3' bias, say like SMART-seq2. You will tile the entire transcript, but you won't actually know the exact permutation combination of exons that are present in your transcript. With ONT, you're going to really sequence it end to end, thereby getting full-length isoform transcript information per cell. So I'm not going to go into the details of the manuscript that Caroline already mentioned, but we wanted to ask a few questions using the scissor-seq technique. So first of all, we looked at 2 different brain regions, the prefrontal cortex and the hippocampus, from a postnatal day 7 mouse, so which is a pretty young mouse. And we asked within a brain region, say the hippocampus, do different cell types and subtypes differ in each other from in terms of their isoform expression, and to what extent? And we found, like the NSMF example, this happens quite often. And depending on whether you're looking at, a neuron versus non-neuron split, where you see 30% upwards of tested genes being really significantly different in their isoform expression, versus subtypes where the percentage is lower, but it also means that it's probably fine-tuning expression. So we see this quite often. And on the other hand, what we also wanted to ask was, if you're looking at the same cell type, say astrocytes, between the prefrontal cortex and the hippocampus. Astrocytes, oligodendrocytes, these are assumed to be homogeneous because they are kind of the glue that binds neurons together. And so in theory, they shouldn't really differ, but do they, was our question. And we find that that actually happens pretty often. In about 5 to 15% of the cases that we looked at, the microenvironment really does influence alternative splicing, and not just of a single cell type like astrocytes, but all of the cell types in a brain region for a gene can have an entirely different splicing profile than that in the other brain region. So this is what we wanted to do, and we did by looking at like genome-wide profiles. And we extended it a bit further by looking at the spatial transcriptomics as well. So this was one of the first assays that came out, and the resolution wasn't that great, but it was still pretty cool because we were able to, just by looking at the gene expression of spots instead of cells, we were able to separate out brain regions with pretty high accuracy. And because the hippocampus is so rich in neurons, excitatory neurons, we called it like a neuron-heavy region. And this is the choroid plexus, this pink region over here, which is responsible for secreting cerebrospinal fluid, or CSF. And because it's rich in non-neurons, we kind of use these as a proxy for neurons and non-neurons. So we originally wanted to validate our single cell studies or results but these spots were a bit too big, housing about 5 to 10 cells per spot. But obviously that's changing now, so that's very exciting. So what we wanted to see is, can we detect differences in isoform expression? And we could. So here I'm showing a full brain slice of the gene PKM with 2 different isoforms. And you can see that in the choroid plexus, there's absence of a particular isoform and presence of another isoform. And these are very starkly contrasting to each other. When you zoom in further into the hippocampus and choroid plexus, you can really see the difference in isoform expression. And the SNAP25 gene is what Caroline showed, so I'm going to skip over it, but you can see this really cool back to front gradient in isoform expression. All right, so this is what we were very excited by, and this was all great at the P7 time point, but there were a lot of outstanding questions that remained. So we basically wanted to ask, these observations that we're seeing, are they persistent when you add more brain regions? We'd studied this at P7, which is like a pretty young time point, and lots of changes happen as we are developing, right? So like what happens as the mice age? And finally, we wanted to also extrapolate to human, because it doesn't matter what we find in mice if you can't extend our results to human and like try and solve disease-related questions. So I'm not going to go into details about how we optimize single nuclei sequencing, because in the interest of time, but we did have to do some degree of optimization because human tissue usually comes frozen, and we had to figure out a way to get full-length isoforms from frozen nuclei. Anyway, so back to the other questions. Can we study whether these isoform changes are similar or different? What happens as mice grow? So here's the study design. We took 4 distinct time points from P14 to P56, with P56 being the adult time point. We swapped out the prefrontal cortex for the visual cortex, but we took visual cortex and hippocampus for these 4 different time points. And at the adult time point, we added 3 more brain regions, which is the thalamus, striatum, and cerebellum. So we have 11 such samples with 2 replicates each, and we ended up with about 200,000 single cells, which I'm representing in this UMAP. And you can break this UMAP down by like different ages and different brain regions. And then when you squint at it, it's not really necessary, but when you squint at it, you can really tell the differences between, say, region-specific excitatory neuron cell types, which are appearing here, say for cerebellum granule cells, or striatal excitatory neurons, so on and so forth. And you can also see how like this is the oligodendrocyte lineage, where you have OPCs, which are progenitors, going on to mature into mature oligos. And at P14, the amount of OPCs is very, very high, and it kind of goes down as the mice age, which is obvious. So we wanted to see like what are the differences when you look at age, region, and cell type. And additionally, there's the cell subtype aspect, because you can have cell type, which is immune cells, but they can be broken down into multiple subtypes. So what are these axes of variability in isoform expression, and can we sort of like summarize them? So the way I went about doing this is, let's consider this hypothetical gene. Say you have gene XYZ for cell type A, and this gene has 5 isoforms. Let's zoom in on a specific isoform, say the salmon colored one. And what you'll see is that there's going to be different brain regions expressing this isoform to different degrees. So on the y-axis is the percent isoform usage, and they can all be very, very similar, or they can be very, very different. So what we do is we calculate the max delta, or the difference, and we call that the regional variation or variability for this particular isoform. We do the same thing for the other 2 axes. So we do it for age, and we do it for subtype, and we report that as a vector. So you're going to have 3 numbers that are describing the variability in isoform expression for 3 distinct axes for a given cell type. And you do this not just for one isoform, but for all of them. And immediately you can start picking out isoforms that are jumping out, right? So like in the blue one, you're seeing a lot of subtype variability versus in the middle one over here, you're seeing a lot of age variability. So we row normalize these so that they add up to 1, and we represent them in this is called a ternary plot. And what that means is that when you look at any of these triangles, you're going to have region, age, or subtype specificity, and in the middle, you're going to have more or less equal variability isoforms across the 3 different axes. So here's a picture of what the variability looks like for excitatory neurons. And just to reiterate, this would be the subtype variability. So for example, between P56, pyramidal cells in the hippocampus versus a P28 cortical layer 2-3 cell. So they're like really fine-grained subtypes. You can have differences between age and differences between region. And as you can see, things are really, really skewed in excitatory neurons towards subtypes. What I do want to point out is that the center triangle over here, it is, like I said, it's uniform, but it can be either uniformly low or uniformly high. So when we looked at the average variability of the 3 numbers that I showed you in the previous slides, we found that most of them are low, but there is quite a few isoforms that are uniformly high across the 3 axes, meaning that they're really, really variable depending on whichever cell's type, subtype, region, age you choose. So these are really probably fine-tuning and modulating function. So this was for excitatory neurons, but you can do this for all major cell types, and you immediately start noticing differences between them. Like excitatory versus inhibitory, you can really tell that inhibitory is not that subtype specific, but you're seeing a lot of age variability. And say in immune cells, you're seeing a lot of regional variability. So you can stare at these all day and find some really exciting examples, which I don't have the time to go over. But next, what we really wanted to do was zoom into what was causing these differences. So what we came up with was a framework to identify extremely variable, not just isoforms, but exons. And the way we did that was by isolating exons that had 75% more change in inclusion between 2 comparisons. So back to this little UMAP representation, we have regions on the rows and ages on the columns. So what you can do is you can pick out cell types. So in this case, I'm picking out the same cell type across 2 brain regions. And I'm asking, is an exon in there highly variable, as in highly different in its exon inclusion? And if it is, then I report it as having an exon, as having adult brain region specific differences. So I can do the same thing for 2 different cell types within a brain region, but in adulthood. So that's adult cell type differences. You can do the same thing at a developmental time point. So it's a developmental cell type difference. And finally, I can do 2 different time points, but the same cell type, and ask, are there any exons that change over time? So here I have my 4 sort of ways of checking for extreme variability. And here I'm showing you those 4 comparisons as columns and my rows as individual exons. And pink indicates that they are extremely variable in one comparison amongst those, columns. And immediately you'll notice that the biggest one in the middle has to do with cell type specific differences. And when we query what those cell type specific differences mean, they really come from the neuronal versus glial split. So on the right, I have the 2 excitatory and inhibitory neuron types, and oligos and astros. And if you look at this network diagram, there's no connection between the glia or between the neurons, meaning that all of these differences are coming from glia versus neuron. And we saw some really exciting examples. I'm just going to highlight one. So one is neurofasciin. It's, again, a synaptic gene. We really like the synapse, but it's expressed in both glia and neurons. What is different, though, is that there's 2 exons that are highly, highly expressed in glia in different brain regions. and 2 exons that are highly expressed in neurons, but not in glia. And when you look at the protein structure, they form the fibronectin 3 type domain, meaning that they're highly repetitive structures, and depending on which domain is present versus absent, it changes the proteins that they interact with. So it's not just like them acting in a vacuum. They're going to be interacting with a lot of other genes. So that was category 1. Then there's other categories. In here, what you see is that you have adult cell type specificity accompanied by brain region specificity. So here's an example. The gene called TEX9, and it has 1 exon that is alternative, but it's alternative to varying degrees depending on which brain region you go to. It can be really low, as low as 10% in the cerebellum versus 90% in the visual cortex. And same for another gene called GRIA. And that is actually, again, like synaptic, I forget, receptor gene, I believe, and glutamatergic receptor gene. And you would expect it not even to be expressed in astrocytes. It is, and not only is it expressed, but it also has very different inclusion patterns depending on which brain region it belongs to. So again, it's like the microenvironment influence that I was talking about in my P7 study. Next, we go into cell type and age specific, meaning that these exons are alternative during development, but they kind of go away in adulthood. So they're transiently cell specific and variable. So in here, with the JAK-MEP2 gene, you have an exon that is present in about 70% of visual cortex excitatory neurons. But when you go into the hippocampus, that number varies drastically depending on what age you're at. The next category is development only, meaning that these change over time, but I'll get to them in just a minute. There's just a side note I wanted to go over. So we looked at all of these EVEX extremely variable exons and their properties, and a couple of things that we noted that were interesting were that these exons in E3 and E5, meaning the ones that are cell type specific, are shorter. So we think they are related to microexons. And the development specific ones, so the ones in yellow, are actually more highly coding than non-coding, meaning that these are transient. They only occur during development, but they definitely play a role in protein coding and doing some sort of developmental things that are crucial for like mice survival. The next thing we wanted to look at is the translatability to human. So we looked at these extremely variable exons, and especially the cell type specific ones, and asked if they were also cell type specific in human. And turns out that not the developmental ones, because we have adult human tissue, but the other ones that are cell type specific are indeed cell type specific in human, but the converse isn't always true. So in 40% of the cases, we find that the human cell type specificity is mirrored in mouse, but there's about 60% of exons that we identified as having cell type specificity, just like glia versus neuron in human. But in mice, that is mostly constitutive, meaning that they're no longer alternative, meaning that somewhere along evolution, humans decided to bring in more isoforms. That's kind of what I said in the beginning of my talk, probably to increase functional specificity. All right, so as promised, the last category over here is the developmental only category, which means these exons are changing over time. So we're going to do a throwback to P1 cerebellum, so really young mice 1 day after birth. And here's a plot of isoform expression for a gene called BIN1. The BIN1 is very important in Alzheimer's disease. And what we see is that there is a very specific neuron versus glia phenotype. So these exons that I'm marking in orange are usually included in neuronal cell types and mostly excluded in glial cell types. So we started calling these, like within the lab, is like the neuron versus the glia isoform of BIN1. And when you look at this newer data, you'll see that at P14, you have the neuronal axon in visual cortex as well as hippocampus, and the glial axon at P56, which is actually kind of conserved throughout. What is interesting is that at P21, you again have the same neuronal pattern of BIN1, but in the hippocampus, that pattern seems to kind of go away. There's a whole bunch of transcripts over here that are not expressing the green, these like alternative exons. So it's starting to take on almost like a glial cell identity. And then when you go into P28, it almost entirely disappears. So what it means to be a neuronal BIN1 isoform doesn't mean that anymore at the P28 time point. And that's also somewhat, not entirely, but somewhat true for visual cortex excitatory neurons. And then at P56, it bounces back. So this is truly transient in terms of isoform expression. And it really challenges the notion of what it means to have a cell type identity. So we're finding this not just in BIN1 as like a unicorn example, but consistently for many exons. And we think that it could do with something called the critical period of development in mice, which is somewhere between the ages of P21 and P30, which is exactly when we're seeing it. But that's super exciting, and we'd love to explore this further, but one thing at one time. So to conclude, we looked at broad, isoform variability patterns across different cell types, and we find, I didn't show you, but about 70% of genes do show isoform variability, not just along one, but along multiple axes. And to wrap up the second half of the extremely variable exon bit, what we found is that the most prevalent changes obviously come from cell type specific differences, both in development and in adulthood, and they mostly come from neurons versus glia. That is kind of not that surprising. The other thing we find is that adult brain region specific differences also exist, similar to what we found with the prefrontal cortex at P7, but they're usually accompanied by cell type specific differences. And lastly, we see developmental differences, which are pretty transient. They occur at P28 or P21, and they're very interesting. So I skipped a lot of things. I wish I could hold you guys hostage for like an hour to go over all of the details of this new work. But since I couldn't, feel free to go check out the preprint. And I'd like to thank the organizers of this webinar for having me here in the first place, for you guys for coming. My ex-former lab and collaborators, and my current PI for being very nice to me and letting me continue my graduate work even after I joined her lab. With that, I'm